Okay, so there are lots of different helminths that humans are infected with, and humans are still infected with these. This is not to scale, unfortunately, but if you take a parasitology class in like medical school or in graduate school, the characteristic of most parasitology classes is really disgusting pictures of worms coming out of people in various ways. So I'm not going to do that to you, but I will try to explain a little bit about the scale of these things. Schistosoma mansoni, which is one of the most important pathogens in humans, we don't talk about it a lot, but it is, after malaria, the #1 daily associated burden of disease for parasites. And it is very small. So the worms are visible to the naked eye, but they look like maybe like a very, 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 very tiny piece of rice, like a very skinny piece of rice. This is in contrast to something like Ascaris, which is, this lives in your liver. We'll talk about these in a bit more detail, but Ascaris, which is a roundworm, which lives in your intestine, and that looks like a big giant worm. This can be feet long, half an inch to an inch around, and you can have dozens of them in your intestine, and they can be quite large, and that's usually where you get a lot of the gross pictures. And then there's various sort of sizes in between these here. The thing about worms that really make them fascinating, that I think make them fantastic for modeling targets from a within host perspective, is they have these incredibly complex life cycles. They make malaria look like kindergarten in terms of life cycle complexity. And they're just, they're bigger pathogens with bigger genomes, and so they have a lot of nuance to them. And so I'm going to take you through 3 different life cycles. I'll try to do it very quickly, but just to show you the breadth, all of these are parasitic helminths. They're all worms of one kind or another, but they're very, very different. So this is schistosoma. Like I said, this is a medically important disease. We'll start the life cycle by the excretion of the eggs from the definitive host, the human, who will excrete them either through the urine or through the feces. And it depends on which species of schistosoma that they're infected with. You can see there's 4 species here, sort of 3 major types that can be secreted either, and depending on the species is how the eggs are excreted. If the eggs get into fresh water, they will hatch, and they'll release these little swimming forms of the parasite called Myricidia that look for freshwater snails to infect. And then they have a whole life cycle in the freshwater snails that will then release the human infectious form, which is called the Cercaria, which are just swimming through the water, and they look like this, like little torpedoes. And if you're wading in a freshwater river, lake, somewhere where this is endemic, and you've got these snails, these will burrow into your skin. They'll lose their tails. They'll burrow through your tissue and get into your bloodstream. This skips a really critical part of the life cycle where they go to the lung, but then they eventually end up in the portal vein of your liver, where they sexually differentiate and they become males and females, and they begin mating and laying eggs that will be, again, released out either through the urine or through the feces. A big determinant of where this parasite is, because this part of the life cycle is so productive, is where the snails are. So we do have the snails in the US, for those of you that are interested, as far north as Tennessee, which is where I live, but we have not had sustained transmission of schistosoma ever recorded. Hookworm is one of the sort of large worms that you probably think about. And here, humans secrete the eggs in their feces. There's no intermediate host. The eggs hatch and form larvae in the soil. So they're just wandering around in the soil. They're very tiny at this stage. You don't really see them. And you're walking around without your shoes on, because it's hot, maybe. And they will penetrate through the skin of your foot usually, and penetrate into the circulation, and they'll go to the lung, and they'll develop in your lung, and they'll become kind of big in your lung, and they'll grow into the airways of your lung, and you'll feel it. And you'll often feel it probably while you're sleeping, and so you'll cough, and you'll cough the worm up, and then you'll swallow it while you're sleeping because you don't realize that you're coughing a worm up. And they're still relatively small at this point, and that's how the worm gets into your gut. And there, it's called a hookworm because this is what its mouth looks like, and it's going to attach itself to the wall of your intestine, and it's going to feed off of you for the rest of your life. These are also very big worms. They can live in you 10 to 20 years. There is a guy who is selling, we'll talk about it a little bit later, but there's a guy that's selling eggs from his own Necator infection and to help you modulate your allergy. We'll talk about why it modulates allergy. So he harvests his own eggs and he'll send them to you in the mail, and you can infect yourself with Necator to help control allergy. Okay, and then the last one we'll talk about is tinea. So this is a tapeworm that is associated really with pigs. We do have this in the US. We have it in Central and South America quite a bit too. It's mostly New World. And so here, humans, the definitive host again. With tapeworms, the reason they're called tapeworms is they get very, very long, and then they start segmenting at the distal end of the worm, and then the segments break off, and those are the eggs. And so you'll excrete them through the feces, and they'll end up in the ground somewhere, and a pig will come along and root around in the feces because that's what pigs do, and they will eat the eggs. And in the pig, these will hatch and form cysts. And these cysts will go into the pig muscle, and then you'll have a nice sort of undercooked pork meal. The cysts are pretty hardy. They survive a fair degree of cooking. You'll eat them, and then they'll hatch into your intestine. And that's what a tinea looks like, and they'll attach to the side of your intestine. This is a major cause of disease because sometimes the pig isn't the intermediate host. So in areas where there's not great plumbing and access to clean water, human feces can get into the water supply. And in that case, you skip this stage and the humans eat the eggs. And the cysts, rather than going into your muscles, go into the human brain preferentially. And so this causes a disease called neurocysticercosis. It's the #1 cause of a lot of neurological sort of epilepsy-like conditions in Central and South America because the brain will just become riddled with these cysticerci. And it's very hard to treat because if you mount an immune response against them in your brain, it causes even greater problems. Okay, so the immune response to helminths, despite that crazy diversity in life cycle and size and biology, really, is sort of all convergent on a response that we call the Th2 response. So just like we've been talking about, there's still an innate response portion of this where you have lots of different innate receptors, including TLRs, that recognize secretory products from the worm. So if you just take the worm and you put it in a dish and you put it in some liquid, and you take that liquid and you put it on cells, you'll get a Th2 response. You can inject it into people, you'll get a Th2 response. This becomes important later. So it's something that they're secreting. It's thought generally to be sugars and lipids, which fits with other pathogens as well. How this gets translated into a Th2 response is much less understood than it is for the Th1 response. It's a more complicated signaling. It seems to sort of involve suppressing the Th1 response at the same time it's trying to activate aspects of the Th2 response. So that's why this is all a little bit confused. But you can just think of it as downstream of innate receptors recognizing secreted components, largely non-protein probably from the helminths. Okay, so then that's going to happen in dendritic cells and other antigen-presenting cells. So remember, the worm is not in the cell because the worm is much, much bigger than the cell, but the cells are picking up little pieces of the parasites that they're shedding off. And then they're going to go talk to T cells and find T cells that are specific for them. And because they've been activated in a distinct way from TB or malaria, they're going to turn that T cell into a Th2 T cell rather than a Th1 T cell. And that Th2 T cell is going to mount a whole different type of response because the goal here is not to kill cells because the worm's not in the cell anymore. So it's going to recruit all these other cell types that we haven't talked about much, eosinophils. It's going to differentiate macrophages to take on a different metabolic life cycle. It's actually going to induce a lot of Tregs, because at this point, you're dealing with this very large pathogen that if you go, if you picture a worm that's this big and a cell that's this big, and you go punching at it with a bunch of inflammation, it's not going to do anything to the worm. It's going to cause you a lot of problems. So you're really trying to tolerate the worm, also get rid of the worm, but do it in a way that doesn't involve the massive scale inflammation that you would for a Th1 response. Again, the cytokines that are associated with this, IL-4, IL-13, IL-5, these are also all the cytokines associated with allergy. So allergy is the bad side of the Th2 response, and we'll talk briefly at the end about their relationship with Helminths. So these eosinophils are these granulocytic cells that have a lot of toxic chemicals that they store up and make into little vesicles, and that they can release when they're triggered by an antibody that's specific for an antigen, or in a particular cytokine environment. And so they're going to get recruited to the site of the worm, and there is going to be some attempt to damage the worm itself. There's also going to be an attempt to potentially attack the epithelial layer where the worm, particularly for these worms that are in your gut, are living, to try to shake them loose. And then also to damage the epithelial layer to allow fluid to rush in, which again, normally you don't think of as very good at fluid rushing into your gut. But the goal is to actually try to flush the worms out when they're these gut worms. There's a lot of cross-regulation now with other bacteria and that are in the gut. And so this is a really hot area of research right now with how worms affect your microbiome components. They're living in the same spot. They're causing all this immune regulation. And in a way, they kind of think of them as two sides of the same coin, in that for most of human history, we've actually just lived with worms for most of our lives. So you can kind of think of worms as another component of the microbiome, an unpleasant one, and one that actually cost us in terms of our health, but potentially also try to evolve some symbiotic benefits for us. And so a lot of the same mechanisms that a helminth, in this case H. polygyrus, does to drive tolerance of its existence in the intestine is mirrored by the same tolerance mechanisms that are induced by certain metabolic components of the microbiome. And so there's this weird convergence of these giant worms with these tiny, tiny bacteria that are both almost commensals, where we think of as the microbiome as generally harmless or even quite helpful. We think of the worms as pure pathogens, but again, they probably have had some positive roles for us in our evolutionary history. And it brings up this idea of tolerance versus resistance. Tolerance is where you decide to, as an immune response, teleologically live with something, make the best of it, get what you can out of it, but you're not going to get rid of it. Versus resistance is where you're going to go all out and try to get rid of it, but it might come at the expense of a lot of tissue damage. And so there are certain situations in which tolerance is maybe the healthier approach from a tissue perspective. But ultimately, almost all the worms I just described to you are bad for us at some level. If you have one roundworm or one hookworm, you're probably fine. And if anything, you might be a little bit better off, which is why people are selling their eggs on the internet. But once you get high worm burdens, and if you're a child, in particular, a growing child, the malnutrition that can be caused by both intestinal worms and even by schistosoma, because they're eating a lot of your blood, it can be incredibly detrimental developmentally. And so when we think about the effector responses that have evolved to at least limit worm burden, it's all downstream of this Th2 cell. So IL-4 and IL-13 and the downstream cytokines are going to drive all of these different distinct effector mechanisms, including eosinophil, which are going to actually try to attack the worm. We'll see a picture of that in a slide or two. And then something called the weep and sweep response, which is this idea of opening up the epithelial layer into the gut, allowing fluid to rush in, and basically just mechanically forcing the worms out because they're in this wave of liquid. Obviously, this isn't super pleasant from a host perspective, but if it gets rid of the worms, it's a very effective outcome. This just shows that from a slightly different angle. When we're thinking about these gut worms, you have a lot of immunity all along your gut. Part of that's to deal with the microbiome, part of that's to deal with the fact that you're getting all these food antigens, and then part of that's to deal with worms. And so you have lymph nodes that are sitting right under your gut where all of this can be mounted. So you're going to get the activation of these Th2 cells that are going to recruit all these cells to the epithelial layer. You're also going to get activation of a particular type of B cell to make a particular type of antibody called IgE. And we talked a little bit about IgE yesterday as the allergy antibody. The reason it's the allergy antibody is it's because it's the worm antibody. So if you have something bad, like IgE is the allergy antibody, it probably has a good component. Evolution wouldn't just make something so that we had allergies that wouldn't be particularly useful. And so IgE as an antibody can bind to antigens on the worms, and it will recruit the eosinophils to dump their toxic components onto the worm surface. And so that can cause damage to the worm surface and prevent them from thriving or replicating. So unlike a lot of the other infections that we've been talking about, there's not sterilizing immunity to worms that we've been able to show in humans or even really in animals. But there's some, like we were talking about for malaria, where if you're exposed long enough and you have the right immune makeup, you can delay your time to reinfection. And this is just one study I cite here where they've analyzed people's IgE levels at a certain time when they surveyed them. Then they treated everybody for worms. So there's lots of good drug options for many worms. And then they looked at their egg output over time relative to their baseline egg output. And the people that started out with the highest IgE levels had the lowest egg outputs. This was for a schistosoma study. And so this repeated exposure led to some immune mechanisms that led to better control. There's a really famous study for schistosoma at a car wash in Africa, where the cars are actually washed in a lake that's infested with schistosoma. It's Lake Victoria, so it's not a lake that they could clear of schistosoma because it's too big. And so they followed the car washers for years, and there were car washers that could make these really potent antibody responses, and they had the longest interval between reinfection, but everybody got reinfected and would have to be treated again. Okay, so I'm going to close there. I actually kind of did it on time today, and just leave you with 3 points that we can talk about. We'll have a little break, and then we'll come back for our half-hour discussion on all the complex pathogens. So parasitic helminths really represent a major evolutionary burden on human development in areas where helminths are still very endemic. It comes at a very high human cost. There's this entire arm of the immune response that's dedicated to worm infections. But in many populations, it just fails to be engaged. So if you grow up in environments where you've been lucky to have access to public health interventions, mainly to deal with clean water and sewage, you're not going to be exposed to worms. And so you have this whole immune system that has nothing to do, that has evolved for hundreds of millions of years to try to get rid of worms. And so there's a very prominent theory that has many forms, but it gets grouped under the idea called the hygiene hypothesis, that our development of allergy and asthma is really a sort of frustrated immune response trying to figure out something to do. And so rather than attacking a worm, it attacks a little bit of pollen dust that you inhale into your lung inappropriately. And so there's a lot of thought that if we just kept worms at sort of a low level, a healthy level that we can all deal with, particularly as an adult, we could actually get rid of allergy and atopy and asthma. And that's this guy that sells his Necator eggs on the internet had profoundly debilitating asthma. This is like a Radiolab episode if you ever want to hear about it, but there's been clinical trials on this as well. But he went to an endemic, a village where Necator was endemic. He walked around without his shoes on. He got an infection, and he claims that it's cured his asthma, and he sells his eggs to other people. There's a lot of work on this to try to obviously come up with drugs that would mimic the effect of having a worm infection, kind of like an adjuvant, without actually having to have a worm infection.